Hello students, welcome to your botany class once again. And today I would like to talk with you the last process of protein synthesis, that is translation. In the previous classes we have discussed that protein synthesis consists of transcription, translation. Transcription is the synthesis of mRNA from DNA, and translation is the process by which the genetic information that is present in mRNA is translated into proteins. So today we will discuss in detail about the process of translation. Okay, so translation is the final step or is the final process in protein synthesis, and it is the process by which various amino acids get arranged in sequential manner according to the information provided by the codons on mRNA, forming polypeptide. Or in other words, there is a change in language during the process of translation. The nucleotide bases present in the mRNA is translated into the sequence of amino acids forming a polypeptide chain, and there's codons on the mRNA, that is the sequence of bases on mRNA, or the codons on mRNA determines the sequence of amino acids in a polypeptide chain. And so the process of translation takes place in the cytoplasm on the ribosomes. And these ribosomes, in case of prokaryotes, there is no nuclear membrane which separates the genetic material or the DNA and the cytoplasm. There's no nuclear membrane which separates it. So mRNA gets transcribed from the nucleus, or not from the nucleus, from the DNA, and then it's directly taken to the cytoplasm for the process of translation. While in case of eukaryotes, there is a clear, definite nuclear membrane which separates the cytoplasm from the nucleoplasm, or where the genetic material is present. So transcription in eukaryotes takes place in the nucleus, while translation takes place in the cytoplasm on the ribosomes. Okay, so the process of translation begins with the activation. Before the process of formation of polypeptide chain takes place, the amino acids has to be activated. The amino acids are present in the cytoplasm in the inactive state. So overall, the process of translation involves the activation of amino acids, the attachment of activated amino acids to tRNA, then the formation of polypeptide chain. That means which involves initiation, peptide bond formation, chain elongation, and termination, and in the end, the modification of the newly synthesized polypeptide chain. So these are certain steps that are involved in the process of translation. Okay, firstly, the activation of amino acids in the cytoplasm. Okay, as I've said, the amino acids are present in the cytoplasm in the inactive state. So, in order for these amino acids to be arranged in sequential manner by peptide bond formation, forming a polypeptide chain, they have to be activated. Okay, so activation of amino acids is brought about by the enzyme amino acyl synthetase by combining with ATP. It's an energy requiring process. So, forming amino acyl adenosine monophosphate enzyme complex. The union of amino acid with ATP forms amino acyl adenosine monophosphate enzyme complex carried out or catalyzed by the enzyme amino acyl synthetase in the presence of magnesium ions, releasing pyrophosphate, and this pyrophosphate further undergoes hydrolysis, releasing energy and inorganic phosphate. So that is the first step during the process of translation, activation of amino acid. So in the second step, this attachment of activated amino acid to tRNA, attachment of activated amino acid to tRNA. So now this enzyme bound amino acid, enzyme bound amino acid, that means amino acyl adenosine monophosphate enzyme complex joins to the 3' end of tRNA, forming amino acid tRNA. So the previous classes we have discussed about the structure of tRNA, where a particular stem in the tRNA has a 3' end and the 5' end, and the 3' end ends is CCA, where A is the end where it receives the amino acid. So this amino acid joins to the 3' end of tRNA, and the enzymes and adenosine monophosphate, they are released. So now this amino acid enzyme complex, or for that matter in specific, the amino acid gets attached to the tRNA, specific amino acid get attached to specific tRNA, and as such, they are now taken to the ribosomes for translation to take place. And these ribosomes are the sites of protein synthesis or the sites of assembling, which acts as the assembling factories. So now, after being activated and after getting itself attached to this tRNA, this complex is taken to the ribosomes, and where the process of formation of peptide bond or the polypeptide chain takes place. Okay, so now the formation of polypeptide chain takes place in 3 steps: initiation, then peptide bond formation or chain elongation, and termination. Okay, so before the peptide synthesis of the polypeptide chain takes place, the prior requirement is that there has to be formation of initiation complex. So we have discussed that these ribosomes are the sites of protein synthesis, and you have learned that these ribosomes consist of 2 subunits, large subunit and small subunit, and prior to protein synthesis, these ribosomes remains in dissociated state. When protein synthesis is not taking place, they remains in the dissociated state in the cytoplasm, but they come together when protein synthesis is about to take place. So these ribosomes remain in dissociated state, then they come together. So before initiation start, there has to be formation of initiation complex. So how is the initiation complex formed? Okay, so in the first step of formation of initiation complex, the small subunit of ribosome joins to mRNA, and we know that the mRNA is transcribed from the DNA during the process of transcription. Then they join to the tRNA. They join small subunit and mRNA join to the tRNA. So this tRNA has transfer RNA has a specific amino acid attached to its 3' end. And you know that during the study of genetic code, we have learned that there is an initiation codon known as the AUG. And in all kinds of protein synthesis, initiation begins with the codon AUG. So this AUG codes for the amino acid methionine. So the initiation amino acid is the methionine. So the first amino acid to enter into the process of protein synthesis is methionine. So this tRNA brings in the first amino acid methionine, which is coded by the initiating codon AUG. So the small subunit combines with mRNA and then joins with tRNA, having methionine at its 3' end, forming the initiation complex. The mRNA and the tRNA pairs up in such a way that the AUG, the initiating codon, forms hydrogen bond with the anticodon of tRNA. So the anticodon on tRNA, which greets the codon and mRNA, they form hydrogen bond, bringing the initiating amino acid, that is methionine. And in prokaryotes, the methionine, the initiating amino acid, is formylated, while in case of eukaryotes, it's methionine only. Okay, why is it that in prokaryotes the methionine is formylated? It is formylated in order to block the peptide bond formation at the amino group of methionine. So the methionine is formylated to block peptide bond formation at the amino group of the amino acid methionine. That's why it's formylated, it's protected. Okay, so now this initiation complex, which consists of the small subunit of ribosome, mRNA, and then formylated methionine, or tRNA having formylated methionine, then now comes and join to the large subunit of ribosome. Okay, so the large subunit of ribosome has 2 specific sites. The large subunit of ribosome has 2 specific sites called the P site and A site. The P site is also called the peptidyl site, and the A site is called the amino-acid site. So the initiation complex formed by these 3 units now come and join the large subunit, form now, and it's totally ready for formation of polypeptide chain to be initiated. Okay, so now this unit comes and join with the large subunit in such a way that the first tRNA having formulated methionine in case of prokaryotes, and in case of eukaryotes is methionine only, comes and occupies the P site of large subunit. So in such a way that this tRNA having formulated methionine at the 3' end comes and occupies the P site, the peptidyl site, and the A site of the large subunit remains vacant. It's free. So it's ready to receive a new tRNA having a new amino acid. So this the entry of the tRNA having methionine at the P site is in such a way that it complements with the AUG on mRNA, which forms hydrogen bonding with the anticodons on the tRNA. And now this A site is ready, it's free, it's called the amino acid site because it's ready to receive a new amino acid or new tRNA having a new amino acid. Okay, so now there are 20 amino acids, and as such, there has to be 20 specific transfer molecules. There are 20 amino acids in the biological world, and as such, there has to be 20 transport vehicles, or there has to be specifically 20, at least 20 tRNAs to transport the 20 amino acids. And also, this reaction is catalyzed by the enzyme amino acyl synthetase. As such, there has to be at least 20 types of amino acyl synthetase to catalyze the different types of amino acids combining with the tRNA. Okay, so now with that, initiation is started. Now the initiation is ready, it's ready, initiation has already begun, and the next step in case of translation is peptide bond formation and chain elongation. The next step is peptide bond formation and chain elongation. Okay, so the process of peptide bond formation is the combining of the amino acids. You know that the amino acids, which are the monomeric units of polypeptide chain, they are joined together by peptide bond. So our concern is with that during the process of translation, the protein or polypeptide chain is formed, and these monomeric units of proteins or polypeptide bonds, polypeptide chain are joined together by peptide bonds. So the second step in case of translation is that peptide bond formation takes place between the amino acids. Let's see how this peptide bond formation takes place between the amino acids. Okay, so in the under this, it involves the binding of tRNA to A site. But the first is binding of tRNA to A site. We have discussed that this, during the formation of this initiation complex, binding to the large subunit, the A site is ready, and the first tRNA having the first amino acid initiating amino acid is bound at the P site, and the A site is ready to receive the new tRNA. So now the tRNA which corresponds, the tRNA having the second amino acid corresponding to the amino acid that is coded by this codon on an mRNA. So in this case, the codon GCU on mRNA codes for the amino acid arginine. Okay, so it codes for the amino acid arginine. So now the tRNA brings the amino acid arginine, which is coded by CGU, and forming hydrogen bonds at its anticodon end. So now this new coming tRNA occupies the A site, amino acid site. So now there are two RNAs having two different amino acids or two amino acids at each end. So one is at the P site having formulated methionine in case of prokaryotes, and arginine at the A site. So how does peptide bond formation takes place? Since peptide bond formation has to take place at least in between two amino acids. So how does it take place is that this arginine, you know that amino acid consists of at least one amino group and carboxylic group acid. So now this amino group, the amino group of arginine carries out a nucleophilic attack on the carboxylic group of methionine. The amino group of this arginine, the amino group of the second amino acid carries out a nucleophilic attack on the carboxylic group of the first amino acid, that is formulated methionine or methionine in case of eukaryotes, forming a peptide bond in between the two, resulting in the elimination of water molecule. So a peptide bond formation bond is formed in between arginine, the second amino acid, and methionine, the first amino acid. So as the peptide bond is formed between these two amino acids, the bond between tRNA, the bond between the peptide bond or the peptidyl bond between the tRNA and the first methionine is broken. It's broken, and as such, this tRNA is released free into the cytoplasm. It's released free into the cytoplasm again, ready to bring a new amino acid again. So now the next step is that now this tRNA has two amino acids, peptide bond is formed, and the next step is that translocation. So now the ribosomes with two amino acids attached to one tRNA at the S side now moves one codon forward. It moves one codon forward in such a way that the amino acid or the tRNA which was previously in the A side comes to the P side, and now the S side is free again. It's free to receive another amino acid, and the third codon comes to the A side. And in case of this, the third codon UAU codes for the amino acid tyrosine. So the new incoming tRNA brings tyrosine and comes and occupy the A side. Again, this tyrosine carries us a nucleophilic attack on the arginine, and then a peptide bond is formed in between again. Again, it moves the ribosomes together with the tRNA moves forward, and as such, the tRNA which is present at the A side comes to the P side. The A side is vacant again, then it brings the new amino acid depending upon the codon on the mRNA. And in that way, peptide bonds formation takes place, and the all tRNA is released into the cytoplasm until and unless it reaches a termination codon on mRNA. So we have studied that there are three termination codons, UGA, UAG, and UAA. So if any of these three codons appears on the mRNA, it gives a signal that translation or protein synthesis has to stop. Okay, so now UAA appears on the A side, and there is no tRNA, there is no transfer RNA which recognize any of this nonsense or stop codons. So the presence of any of these codons on A side give the signal that translation or protein synthesis has to stop, and in that way translation stops, and the bond between the last amino acid and tRNA which is present at the P side is broken, and the peptide bond or the polypeptide chain is released into the cytoplasm. And once it is released, the proteins again, the polypeptide undergoes modification because the newly synthesized protein is inactive. As such, it has to undergo modification of secondary and tertiary folding, giving rise to active form, then formulated group in case of F-methionine in prokaryotes is removed, and then the ribosomes dissociates back again, and then they dissociate back into the cytoplasm. Besides that, this newly synthesized polypeptide chain undergoes modification of phosphorylation and glycoproteins, and as such, they becomes only active after modification. Okay, so when we have discussed that the ribosomes are the sites of protein synthesis, so ribosomes may be freely scattered in the cytoplasm. In case of prokaryotes, or in case of eukaryotes, they can be freely in the cytoplasm. They can be scattered in the cytoplasm, or they can be present even on the endoplasmic reticulum. Okay, so when protein synthesis takes place on the free ribosomes, the newly synthesized proteins is released freely into the cytoplasm of the cell. And so they take part in the formation of enzymes, cellular components like mitochondria, chloroplast, or they can be carbohydrate molecules are added to it so that it forms glycoproteins. And when protein synthesis takes place on ribosomes, which are attached to the endoplasmic reticulum, that is a rough ER, they enter into the lumen of ER, and then from the lumen of ER, they goes into the Golgi apparatus, and in the Golgi apparatus, they get back as hydrolytic enzymes forming vesicles, and whereby they form lysosomes. So this is how protein synthesis takes place in case of prokaryotes and eukaryotes. Of course, there are certain differences between this. during the process. So the one difference is the formation is the initiating methionine, initiating amino acid that is formulated in methionine. When in case of eukaryotes, it is methionine. Okay, so translation results in the formation of polypeptide chain. With this, I conclude the process of translation and the process of protein synthesis. Thank you.